All right, take two on this video now that I've got my terminologies correct. I want to get back to this world of digestion. We've talked at length about the idea that we have to break material down that we eat. That started out first as food, and then it became a bolus. And now as it comes into the duodenum, it's referred to as chyme, C-H-Y-M-E. So we have to look at all the chemistry that is going to take place as the chyme enters the intestines, which is going to be all about absorption. When it comes to the intestines, there's not a lot of chemistry. The chemistry is all up in here. So that's why I decided, let's take this pancreas, split it out, take a little bit more time with it, because I'm going to throw a lot of chemistry at you. Remember, chyme coming in here is very low pH. It's very acidic. It's got a handful of different enzymes that are breaking down those biomolecules that are the major ones: proteins, lipids, carbohydrates, and nucleic acids, both RNAs and DNAs. The bile is going to go to work even further on, even further on the lipids, on emulsifying those, and we're going to have some things that are going to come in now from the pancreas that will help with the digestion and ultimately the absorption of the lipids themselves. Here we go with a look at the pancreas, zooming in a little bit more. We see our common bile duct. Here's our pancreatic duct. See, I said it went all the way out to the tail. We've got a little shunt over here that we won't talk much about. But we've got these groups of different colored, when we look at this, cells. Some of them are endocrine, some of them are exocrine. The endocrine is going to secrete directly into blood vessels. Just like the liver did, it's got multiple functions. The pancreas is going to play a role in regulating blood sugar, as well as helping with digestion down the line in the intestines. So I think where I want to start out with is looking at these acini, or these acinar cells, and look at what the enzymes are that are going to be secreted in, along with the bile duct, that common bile duct, and then we'll get to the endocrine to finish this up. So here we go. Here's where we were. I finished off that last video talking about bile being the emulsifier of fats, breaking down these big blobs of fat into a lot of tiny blobs of fat. And now we can get into what the pancreas secretes. Lipase, or lipase, as some people say. We're going to do the same thing that we saw up here. Lingual was from the saliva, pancreatic is from the pancreas. I think that's pretty much straightforward. So these are going to work on breaking down the fats that you have taken in. Then amylase. Oh, look, salivary amylase. Now we have amylases from the pancreas as well. So we really start to build this list. Ribonucleases. Ribonucleases will break down RNA into the individual nucleotides, the A's, the U's, the G's, and the C's. And then close to that are the deoxyribonucleases. I have to change my formatting because the slides are getting too full of chemistry. And instead of breaking down RNA into A's, U's, G's, and C's, it is going to break down DNA into A's, T's, G's, and C's. So what I think you can see that we've done by the time we get into interacting the chyme, the material that's come out of the stomach, with the pancreatic juices, we've got a massive chemical breakdown, a chemical attack on what it is you just ate. Then there are zymogons, one of those other terms that it sounds like I probably made up, and I didn't. Zymogons are a group of molecules, a big group of molecules. And the ones we're going to look at that are going to be pancreatic in origin are all going to have to deal with breaking down proteins. You might be able to think for a minute about what would the problem be. Why would we call it not just more proteases? Kind of like pepsin. Well, here's one of the problems. If you build and secrete a molecule, an enzyme that's going to break down proteins, and you secrete it or build it within yourself before you secrete it into the lumen of the duodenum, you might tear apart your own proteins. So what a zymogen is, and this is kind of a family of molecules, is it's an inactive enzyme that's going to become active after it's been secreted. So follow me on this one. I may have to do a little drawing with this. Here's what we're dealing with when it comes to the world of zymogens in the pancreas. There are these three big zymogens: trypsinogen, chymotrypsinogen, and procarboxypeptidase. All of these are inactive. And as I put it there in really tiny font, they'll all become active once they're in the duodenum or the duodenum. So how do these things all work? What's the role of all these? And I'll take them one by one by one. Let me just give you this up front. These two, the chymotrypsinogen and the procarboxypeptidase, are going to become active due to trypsinogen becoming active. If that makes sense, but follow along. I hope it will. Trypsinogen becomes trypsin, so I guess I could do an arrow like this. Once it comes in contact with enterokinase, enterokinase is something that is secreted. It really doesn't do anything in the intestines, the small intestines. So enterokinase is sitting in the small intestine. Trypsinogen gets secreted in. Suddenly it becomes trypsin. So trypsinogen has been activated. Now what's trypsin do? Well, trypsin is going to start to break down proteins, kind of random proteins here and there that you've ingested from your can of sardines. But it's also going to do two really important things, and it applies to these next two bullet points. One is that it converts trypsinogen, or I'm sorry, I just went back one bullet point, chymotrypsinogen to chymotrypsin. So I put over there, it's done by trypsin. It's not done by the enterokinase. Now what's chymotrypsin do? It breaks down proteins. So suddenly in the lumen of the small intestine, we're activating this massive attack against proteins to turn them into amino acids. And the same thing is going to happen right down here. Our procarboxypeptidase becomes carboxypeptidase, so we've essentially knocked off the pro here. We've knocked off the gin here, and we've done the same thing with the ogin here. Little tiny changes in the spelling, in the name, indicates a big change in what it does. Inactive on the left, active on the right. Not attacking anything on the left, attacking everything on the, well, shouldn't say everything, attacking proteins on the right. So, trypsin itself, I think as I mentioned before, along with carboxypeptidase and chymotrypsin, will all mount a huge attack on the proteins that you just ingested. So by this time, if I was to back this up, work with me on just a couple of slides. Now these zymogens, we've got a few more down here. Look at the chemistry. This is why I don't care if we take a little bit of extra time on the pancreas. It's just one organ. Can't be that important, but it is. It really, really is, and there's a lot of chemistry, and that can confuse a lot of people. So, as we move back, forward through these zymogens, all of this has been dumped into the lumen of the duodenum, and now we can get to work on finishing up the digestion in the three different regions, the three parts of the small intestine, and then we can look at absorption, which won't take long in the next video. But I can't ignore one last thing with the pancreas. I mentioned that the pancreas has endocrine cells and exocrine cells. Exocrine is all that's secreted into the duodenum. Endocrine will be secreted into the circulatory system, into the blood. Now, this may be very important to some of you. This might have a direct impact on some of your lives. I know that I have family members that are impacted by the types of cells that I'm going to try and introduce to you next. Those cells are called alpha cells, and they're called beta cells. So we have pancreatic alpha cells, and we have pancreatic, there they are, beta cells up here. And there are a couple of different endocrine molecules that are going to be secreted. Insulin and glucagon. Some of you know right away if this is going to be important to you, if it impacts you directly. Some of you, it would be nice if you go, I don't know anything about glucagon or insulin. Fantastic. Myself, I produce the correct amount of insulin for an old man, the correct amount of glucagon for an old man. And what I've done is I've taken this loop. And so let me start out where I like to start out, eating. So eat a couple of Kit Kat bars. All of a sudden my blood sugar has gone up. So suddenly this is a circulatory system issue. My pancreas realizes that, and my pancreas is going to kick out insulin. And what insulin is going to do, this hormone, is going to tell my cells, specifically my adipose cells, and to a certain extent my liver, start pulling up sugar. Try and reduce the amount of blood sugar. Pull that out of there. And that's what it does. And it turns it into glycogen, and in the long term, it'll turn it into fat. So if my glycogen, or if my glucose goes up, my insulin will bring it back down. It's one of the reasons if you eat a really sugar-rich meal, or maybe a couple of candy bars between classes, all of a sudden you find yourself exhausted because insulin pulls your blood sugar back down. Well, those are coming from the beta cells. On the flip side, as I'm riding my bike, or I'm playing with my dogs, or you're playing a 3-on-3 pickup basketball game, you're pulling the blood sugar out of there. Well, what that does is it tells your alpha cells to release glucagon. Glucagon is a sugar. It goes to the liver, turns the glycogen, which is what I mentioned up here. This is how we store, kind of in the short term, our energy, into glucose or blood sugar. So all of a sudden, if I happen to be riding my bike and I had a lot of carbohydrate the day before, what I did is, because I wasn't using it, I was storing it as glycogen. And the next day as I ride, I'm going to start pulling it out and turning it into glucose or blood sugar. So I can regulate this back and forth to make sure my blood sugar levels don't do something like this, but instead they do something like this. It's that homeostasis thing that we've all talked about. So as I mentioned, a lot going on with the pancreas. And here where these, as I get rid of the scribble, these different molecules have come from. Here we've got our alpha cells and our beta cells. Don't worry about deltas and things like that. But around the outside of this group of alpha and beta cells, we have the acini, and this is where we end up getting all of our digestive enzymes and our digestive chemistry that comes from the pancreas itself. So where we have to go next, let me get that scribble off of there, is into the intestines themselves. So we started out here, done that, gone through the stomach, done that, looked at the liver, done that, the pancreas hiding back here, done it. Now we got to look, we got all of that coming together in the chyme. We've got this big long run through the small intestine, a loop up and across and down the small intestine, and we're done. So that'll be in the next video, and that won't take long for me to queue up, I hope.